Let's now look at how different tissues of the developing central nervous system become organized. The neurons of the brain are organized into layers, which are known as lamina. They're also organized into clusters, which are known as nuclei. The neurons within the same nucleus or cluster make similar connections and have similar functions. And recall that these cells are derived from a one-cell layer thick epithelium, and this epithelium spans the entire apical basal surface of the neural tube. There are adaptations that have led to the germinal epithelium producing diverse types of very complex regions within the central nervous system, and all of these regions are elaborations of a basic three-zone arrangement. And this three-zone arrangement is composed of the ventricular zone, the intermediate zone, which is also known as the mantle, and the marginal zone. The ventricular zone is a region that is in contact with the lumen of the neural tube. So this is the opening of the lumen of the neural tube, and on the opposite side would be the outer region or the outside. As cells of the ventricular zone divide, and you can see these cells here, which are the radioglia, and they're shown in the lighter blue color, and they span the entire width of the neuroepithelium. Anyway, as these cells divide, their daughter cells move using them, the radioglia, as a scaffold. So the descendants of the radioglia move to form the second layer, the mantle zone or intermediate zone. And cells from that layer are going to give rise to both neurons and glia. The neurons are going to make connections among themselves, and they're going to send out axons. And that forms that third layer, which is the marginal zone. Eventually, oligodendrocytes wrap themselves around these axons, and that gives the portion of the cells that project into the marginal zone a whitish appearance. So this becomes known as white matter, whereas the intermediate or mantle zone, which has many cell bodies, is the gray matter. The germinal neuroepithelium that's here at the ventricular zone eventually shrinks and becomes the ependyma, which lines the cavities or brain vesicles. And if you recall, the ependymal cells are responsible for secreting cerebrospinal fluid. As we discussed earlier in the semester, this region of the ventricular and subventricular zone will continue to house a population of neural stem cells. This basic arrangement in the neural tube of the ventricular, intermediate, and marginal zones gives rise to the various layers of cells that are in the spinal cord, the cerebellum, and the cerebral cortex. And notice how the radioglial cells, which are shown in blue, span all three layers. And remember that these cells are not only going to serve as stem cells, but that they're also the scaffolds for the migration of neuronal progenitors and newborn neurons. But before we discuss the organization of neurons in the major parts of the CNS, let's take a quick look at these parts and the regions of the neural tube they derive from. Recall that the neural tube becomes divided into three primary brain chambers: the prosencephalon or forebrain, the mesencephalon or midbrain, and the rhombencephalon or hindbrain. Posterior to these, of course, you have the region of the neural tube that will give rise to the spinal cord. And these chambers become further divided into different regions along the anterior-posterior axis. The three regions we'll be discussing today are the spinal cord, the cerebellum, and the cerebrum. The cerebellum, as you can see from the image, is derived from the rhombencephalon or hindbrain. The cerebrum, on the other hand, is derived from the prosencephalon or forebrain. This is a graphical representation of what these regions look like as the embryo develops and what they look like in a human child. As you can see, the cerebellum is located near the base of the brain. Notice also that the most anterior portion of the forebrain, the telencephalon, basically folds so that it appears to encase other portions of the brain derived from more caudal regions of the neural tube. And you can see this more clearly in this image, where it's apparent that the cerebrum, which is derived from the telencephalon, covers the diencephalon, which is a little more posteriorly located relative to the telencephalon, and it also covers the midbrain, which is the mesencephalon. So the more anterior region of the forebrain, the telencephalon, which gives rise to the cerebrum, covers these more posteriorly derived regions. Now that we have a picture of the anatomical organization of the brain, let's discuss the organization of cells of the spinal cord, the cerebellum, and the cerebrum. Let's begin by looking at the development of the spinal cord. And as you can see from this image, that pattern of organization of the three zones, the ventricular zone, the mantle or intermediate zone, and the marginal zone, is retained in the spinal cord. Also in the medulla, which is not shown here, and it's the posterior part of the hindbrain. Now this is a cross-section through the spinal cord. And as you can see, the mantle gradually becomes shaped like a butterfly, and it's surrounded by the marginal zone, which is the white matter. And both the marginal zone and the mantle are going to be surrounded by connective tissue. As the neural tube matures, a groove that's known as the sulcus limitans forms. This groove divides the dorsal and the ventral halves of the spinal cord. And remember from the previous lecture that the dorsal portion is going to receive inputs from sensory neurons, and the ventral portion contains motor neurons. As you saw, the spinal cord has a very simple three-layer arrangement, which resembles the initial arrangement of cells in the neural tube. But in the cerebellum, additional cell migration and proliferation give rise to additional layers. So the cerebellum not only has a ventricular, mantle, and marginal zones, but also contains additional layers, an internal and external granular layer, and a Purkinje cell layer. The outer regions of both the cerebrum and the cerebellum become highly folded, as you can see here. In the cerebellum, the outer regions contain granular cells and neurons with very dense dendritic trees or dendritic arbors, and these are known as the Purkinje neurons. Purkinje cells participate in motor control and learning. Although they can receive input from hundreds or thousands of cells, they are the only cells that emit signals from the cerebellar cortex or outer layer of the cerebellum. One of the critical events that occurs in the region of the cerebellum is the migration of neural progenitor cells to the outer surface of the developing cerebellum. This process gives rise to an external granular layer which is near the outer boundary. These cells of the external granular layer are progenitor cells that proliferate at the outer boundary before migrating backward to create an internal granular layer. As in the cerebellum, in the cerebrum or cerebral cortex, the three basic zone arrangement consisting of the ventricular zone, the mantle zone, and the marginal zone becomes extensively modified. Some neural progenitor cells of the mantle zone migrate toward the outer surface, and they form this new layer known as the cortical plate. And this new cell body rich region, this cortical plate, will gradually give rise to the neocortex, which is a distinguishing feature of the mammalian brain. Because the cortical plate is a cell body rich region, it is also classified as gray matter. The neocortex consists of 6 layers, which you can see here. Interestingly, these layers are not fully mature until mid-childhood. Each of the layers differs from the others in the types of neurons that are present there, the functions of these neurons, and the connections that they make. For example, the cells in the 4th layer are cells that receive output from the thalamus, whereas the 6th layer is the one that sends outputs to the thalamus. We'll discuss the formation of the cortical plate in a subsequent video. In addition to being organized into layers, the cerebral cortex is also organized horizontally or cross-sectionally into distinct functional regions, as shown in this simplified diagram. So each of these portions of the brain controls different functions. Studies suggest that the specification of these different functional regions happens early, prior to the cells migrating out of the ventricular zone. Some evidence for the early specification of these functional regions comes from studies which show that mutations that severely affect one part of the cortex do not have a significant effect in other parts. For example, a mutation in the G protein coupled receptor 56, GPCR56, affects cortical lamination. The region of the brain that is most affected by this mutation is the frontal cortex, while the parietal and temporal lobes appear relatively normal. If the specification of different functional regions happened later in development, and if it was related to the connection between these regions, then we would expect that these mutations would cause widespread abnormalities. Additionally, the experiment depicted here provides further evidence for early specification of different neuronal cell types. In this experiment, specific ventricular radioglial cells, which are the neural stem cells, were labeled. And this was done so that their clonal descendants could be followed during development. It was found that differentiated cortical neurons in specific areas descended from stem cells that are present in similar regions, and that these stem cells were themselves derived from radioglia from comparable ventricular zone areas.